I presented today results from a genetic analysis in survivors of childhood cancer from the St. Jude Lifetime Cohort study. So this is a study on the survivors of childhood cancer, and as we know, cancer is a leading cause of death in children under the age of 14 years in the US, and in 2019, an estimated 11,000 children will be diagnosed with cancer, and roughly 1,200 of them will be dying from the disease. But thanks to the treatment, advances in treatment over the last few decades, more than 85% of children now survive 5 years or longer. And this is a huge increase since 1960s, when only 20 to 30% of children were surviving 5 years or longer. However, cancer therapy have caused some health problems. Some of the problems can be short-term, which can occur during the cancer treatment or shortly after the therapies. But some problems can occur later in the life, and they may not go away, or they may not even show up until months or years after the treatment. And these kind of treatment we call late effects. And these late effects can include secondary cancer, cardiovascular complications, including stroke, infertility, and other conditions. And these are associated with significantly increased risk of mortality, morbidity, and impaired quality of life. So we are particularly interested in stroke, which is a major cause of physical disability and cognitive impairment. And survivors are almost 8 times more likely to develop stroke compared to their sibling controls. And cranial radiation therapy for the primary cancer remains to be the strongest risk factor for stroke in survivors, with a strong dose-response relationship. With compared to survivors who did not get cranial radiation therapy, survivors who got 30 to 50 gray are almost 6 times more likely to develop stroke, and those who got very high dose of radiation, including 50 gray and higher, they are at almost 11-fold risk of stroke. However, there is a wide risk variation in survivors who got cranial radiation therapy. Not everybody who got same level of cranial radiation end up developing stroke, and some survivors who got the same dose, they also get the stroke down the road. So that indicates us a potential role of genetic predisposition in risk of stroke in survivors. So to answer that question, we comprehensively examined whole genome sequencing data obtained from the cutting-edge next-generation sequencing technology from the St. Jude Lifetime Cohort study. So this study is a retrospective cohort study with a prospective clinical follow-up and ongoing enrollment of childhood cancer survivors who have survived at least 5 years or longer and are treated at St. Jude Children's Research Hospital. So using that data, we perform a comprehensive analysis, and our results identified a chromosomal locus on chromosome 5, which were strongly associated with risk of stroke. And in overall, survivors who had a risk allele in that chromosome 5 locus, they had almost 3 times more risk of developing stroke compared to those who did not carry the risk allele. So, and we replicated these results in African survivors from St. Jude Life study. In addition, in survivors of European ancestry in the St. Jude Life, we also observed cranial radiation dose-specific effect for the SNP. So in compared to among survivors who were not exposed to cranial radiation, the SNP conferred almost 1.5 times risk. And in survivors who were exposed to low dose of radiation, which is between more than 0 and then less than 25 gray of cranial radiation, the risk was increased to 2.4-fold. And in survivors who got intermediate dose of cranial radiation, that is between 25 to 50 gray, the risk was all up to 5 times. And in high dose exposed survivors, the risk was almost 3 times. So in summary, so we conducted the first genetic study for stroke in survivors of childhood cancer and identified chromosomal locus which was associated with risk of stroke with 3 times. And we also found potential cranial radiation dose-specific effect for the SNP. And so we are looking forward to independently confirm this association in independent samples, and also working towards functional experiments to understand the underlying biological mechanism behind this genetic association. This is going to help in terms of identifying the high-risk survivors who are exposed to cranial radiation so that we can explore about intervention strategies. But to do that, we need to still do the validation studies and to understand the mechanism behind the genetic association we have identified. Earlier studies have already shown that in survivors of acute lymphoblastoid leukemia, which is the most common type of leukemia in children cancers, so the cranial radiation the treatment has been already removed. So this study is the beginning. So we need to do additional studies and to explore options about whether to modify the treatment strategies or to especially to identify high-risk individuals or survivors for the intervention strategy. For example, the recommendation could be to modify the cardiovascular risk factors because they are already at higher risk genetically for the increased risk of stroke. Discovery analysis included almost 700 survivors of European ancestry from the CNZ lifetime cohort study. And so that included almost 17%, 116 of those had a stroke. And in terms of the risk allele that we identified, almost 50% of those individuals who had a stroke carry at least one copy of the risk allele. In childhood survivors of European ancestry, the common risk allele that we have identified in chromosome 5, so that had a cranial radiation dosage-specific effect, especially among survivors who received intermediate dose of cranial radiation, that is between 25 and 50 gray. For the other groups, the risk was almost 3 times, similar to the overall population. And in survivors who did not get cranial radiation, the risk was almost half, which is almost 1.5 times. One of the late effects that we are interested in is stroke, which is a major cause of physical disability and cognitive impairment. Survivors of childhood cancer are almost 8 times increased risk of stroke compared to their sibling controls. And the cranial radiation therapy for the primary cancer remains to be the strongest risk factor with a dose-response relationship.